So what's the mode of inheritance for C? Let's look at it first. Here you have, this is some red blood cell, which has this H antigen on the surface, right? And then you have here A type of blood, so it has this extra thing on top. So what the extra thing is written here. So N-acetylgalactosamine. So the A has this extra structure on top of the H antigen. And if you have blood type B, so you have the red blood cell, which has this extra thing here that is different from that. So this basic part is the same for all of them, for the O, the A, and the B. But then the A adds the black, was this hexagon? Yeah, the black hexagon. And then this guy has the yellow hexagon. And then if you are AB, the red blood cells have the same basic structure, and they have both this one. So this one would be the B antigen, and also has this one on the same cell. So it has the A antigen. That's how the red blood cells of AB people are. So let me show you now the answer for that. So the correct answer for that is codominance. And some people are like, maybe incomplete. Now this is codominance. So the way we define it as codominance is that there isn't an A or a B. You could have somebody who's AB. So you have the expression of both alleles, the alleles that will encode the adding this A antigen and the allele of adding this B antigen. So because they're not mutually exclusive, you can have both, and one doesn't take over the other, then this one is codominance. Now there's a wrinkle here, which is if you had this, if you had an allele that encodes A, which says you have an enzyme that adds this thing to the H antigen, and you had an allele that encodes O, that's your little I, if you remember from 211 lab, then you have blood type A, because you have an allele that says that you can add this structure to a H antigen. So the relationship between A and O is actually complete dominance. But when we think of blood types, mostly people are focusing on the A and B and the fact that you can have AB. So the blood type 1 is the tricky one. So when you were talking about alleles that encode the antigen A and allele that encode the antigen B, that's codominance. But if you had an allele that encodes the O and another allele that encodes A, then you're blood type A. That's complete dominance. Or if you had one allele that encodes O and one allele that encodes B, you're blood type B. So the B is dominant over the allele that encodes O. You're going to see why in the next slide or something like that. Hopefully you found that very interesting. So you are wondering, how does this molecule get added to the H antigen? And I'll be like, I'm glad you asked. So that is the type of things that I wanted to think about. What's the mechanism that makes this molecule have something added to it? So this one is codominance, and focusing on the A and B alleles, they are codominant. And between the O, or the little I, that's the allele that encodes blood type O, and A, or O and B, that's complete dominance. Okay, so as what's it called, by exclusion, what's the middle one? Just put on the chat. Right, yeah. Good job, Michael. Good job, Miss Sydney. Yeah, so the middle one is incomplete dominance. that's how people thought, initially you'd think, okay, you mix red and white, you're gonna get pink. And like, sure, that makes sense. So the incomplete dominance has to do with a dose. So there is a dose of red pigment here and a dose of white pigment here. And then you mix them and you end up with, so red and white end up with pink. The middle one is incomplete dominance. And then you are going to see examples of how that happens. It's pretty intricate, but I don't want you to actually memorize how it happens. I want you to understand how do you end up with phenotype that is somewhere in between. So far so good?